Hey class, let's talk about cells and taxonomy. If we look at cells, bacteria and protozoa cells are almost always, or life forms are always going to be in a single cell state. Animals and plants are almost always going to be in states of having trillions upon trillions of cells. As we look at cells, there's going to be different characteristics that define the cells. Maybe they're spherical, maybe their shape is polygonal, cuboidal, cylindrical. Depending on the specific cell type, it will have a specific shape. Depending on the cell type, maybe there'll be a cell membrane, maybe there will be a cell wall and a cell membrane, maybe that cell is going to have big pieces of DNA forming multiple chromosomes, or maybe there's going to be one piece of DNA forming a loosely coiled chromosome. Some cells are going to use ribosomes for protein synthesis. Actually, most cells, I should say, use ribosomes for protein synthesis. And then our cells can be exceedingly complex to much more simple in function. All eukaryotic cells, as I mentioned before, have membrane-bound organelles and have a true nucleus. Animals, plants, fungi, and protozoa fall into that eukaryotic cell state. Bacteria and archaea don't have a true nucleus or membrane-bound organelles, and they have very fine structure and can engage in every activity that our eukaryotic cells can, but they are not as complex as the eukaryotic cells. Eukaryotic cells are typically bigger and more complicated, whereas bacteria and archaea are simpler and less smaller, I should say, simpler and smaller. So this is actually more of a question related to the last video they just recorded. What are cell membranes made out of? For those of you who have your PowerPoints handy, you should be able to flip back a couple slides. If you're just watching this YouTube video, I'm going to flip back for you because it's not in this recording. Cell membranes are made of... Here we go. Here we go. Phospholipids. So here's a standard phospholipid, and phospholipids are made or make up the majority of our cell membranes. Cell membranes also have steroids, particularly cholesterol, embedded in them, and every now and again they'll have some proteins and carbohydrates embedded in them. So the best answer here is phospholipid. But technically cell membranes have phospholipids, carbohydrates, steroids, and proteins in them. But the cell membranes are mostly made of phospholipids. Based off these characteristics of the cells and the organisms, the DNA, the ribosomes, particularly the rRNA, the RNA that makes up the ribosome, we can classify and categorize the organisms. Taxonomy is the science of classifying living things. It was developed by Carl von Linné, also known as Carl Linnaeus. He lived from 1701 to 1778. And he used a series of nomenclature, or the assignment of scientific names to different kinds of living things as he was categorizing them, or classifying them. Classification is formally known as the orderly arrangement of organisms into a hierarchy, and identification is formally known as the process of finding and discovering a new organism, or in other words, you identify the organism, and then you put it somewhere in this classification and nomenclature scheme, or you use taxonomy to categorize a new organism that you have found or identified. When naming organisms in a scientific fashion, there are very specific rules that need to be followed. We use old languages like ancient Greek or ancient Latin to name our organisms with a scientific name. A reason we use these dead languages is because they don't change very much anymore. Since most people don't talk in ancient Greek or Latin anymore, we don't have lots of new slang words appearing or nicknames appearing. When you talk about the common name of an organism, different parts of the country, different parts of the world can have different common names for the same organism. But worldwide, there's only one scientific name for an organism. When we use the scientific name to name an organism, it's a two-word name, or a binomial nomenclature. And these two words are made of the genus name and the species names. When we type a scientific name, it must always be italicized. Always put a scientific name in italics. When we print a scientific name, you're supposed to underline it, because it's very hard to handwrite something in italics. Something else that should be emphasized is the genus right here is always going to be capitalized. And then the second part of the binomial name, the species name, is always lowercased. If you're abbreviating a scientific name, the genus is abbreviated with just the first word the first letter followed by a period, and then the full species name is written. So if I wanted to say the abbreviation of Escherichia coli, I would just say E period coli. The different categories of within classification or within our taxonomic scheme are going to be large and small, depending on the category. We have the largest categories known as domains, and then we have the smallest categories, or the most specific categories known as species. We go from largest to smallest: domain, kingdom, phylum, class, order, family, genus, species. And there's a lot of different mnemonic devices that can help you remember this order. So here are two examples. Let's look at a human being. Human beings are categorized as Homo sapiens, genus Homo, species sapiens. But if we're going to be in the kingdom Animalia, we're technically considered an animal, and of those animals, we're considered animals with backbones, so we're going to be in phylum Chordata, and of those animals with backbones, we are mammals, so we'll be put in class Animalia. We are specifically primates. We are a mammal that falls into the category of primates. And then finally, of those primates, we don't have very pronounced tails, so we're going to be in the family Homoodea, and then specifically the genus is Homo, the species is Sapien, so the scientific name for human beings is Homo sapiens. Let's look at another organism over here, Paramecium caudatum. Paramecium caudatum is going to be in kingdom Protista, phylum Celiophora, class Hymenostomia, order Hymenostotia, which is almost identical to class, I might add, and family Parameciidae, genus Paramecium, and then species Caudatum. Paramecium caudatum. As we have gone through the category of naming and categorizing our organisms, we've had the theory or the field of phylogeny develop. Phylogeny or phylogeny is a way of having a taxonomic or categorical scheme that represents how things are related to each other. According to the theory of evolution, when we have genetic information go from one organism to another organism, there'll be small changes over the course of time, small mutations that build up. These small mutations that build up occasionally are good for the organism and cause there to be more and more small beneficial mutations building up over the course of time. Eventually, if enough of those build up over the course of time, we have a new species. The force that selects for these beneficial mutations is called natural selection. Natural selection is the idea that if we have two organisms, one that's healthier than the other one, the healthier organism will reproduce more than the unhealthy organism, and that healthier organism that reproduces more will transmit its genetic information with its small mutations onto more of its offspring and change the population more than the organism that's not as healthy. When categorizing our organisms, we've gone through multiple schemes. Earlier on, Charles Darwin and Ernst Haeckel had proposed that there are just two kingdoms, plants and animals. Later on, Haeckel added Protista, and then he had Monera. And then according to the Whittaker model, fungi was separated from plants and added as a separate kingdom. And these categories were based on structural differences and similarities. Now we use a different system. We use the Woese-Fox system. The Woese-Fox system is based on the ribosomal RNA, specifically for the small subunit of the ribosome, the rRNA sequence that makes up the small subunit of the ribosome. And based on that sequence of genetic information, we have a new categorical system. Why did we use the small subunit of the ribosome? All organisms have ribosomes. To my knowledge, every single organism on the planet, whether it's multicellular or unicellular, has ribosomes in it. And the ribosome accumulates mutations very, very slowly. So as we look at the sequences that make up the small subunits of the ribosome, it's been found that there are separate groups for bacteria, to separate bacteria from archaea, or archaeabacteria. So we have a totally new system that's been proposed. It was proposed in the late 1990s. It's based, and these domains are now bacteria, archaea, and eukarya. And this is all based on the genetic information. We, as human beings, fall into domain archaea, and then there's domain bacteria and domain archaea. So, concept check, guys. Which of the following domains are in the Woese-Fox System of Taxonomy? So this is check all that apply. Are Protista? No. Are Eukarya? Yes. Are Archaea? Yes. Are bacteria? Yes. Are fungi? No. So you should check B, C, and D. Those are the domains in the currently used system of taxonomy. Protista and fungi are no longer considered domains anymore. If you have any questions, please shoot me an email. Happy studies!